Right, I hope everyone can see my screen. Good afternoon, everyone, and good morning to some of you. Welcome to this online workshop, a practical guide to genome-wide association study. My name is Zihao Zheng. I'm a PhD candidate in Dr. Pat Schnabel's lab at Iowa State University. And I left my contact information here on the first slide. So if you have any questions after the workshop, or you need some help with your GWAS analysis, you can just shoot me an email or follow me on Twitter. And also here's the link to my GitHub. So some introduction and objectives of this workshop. So this online workshop is part of the AG2PI online workshop series. So this is workshop number 4. So if you haven't checked it out, please be sure to check out the previous workshop, including the foundations of the computational biology, a collection of topics on linking the genotypes to phenotypes, and especially workshop number 3 last month, the SNP data analysis. So the recording and the slides are all available for these past presentations. So the recording and the slides will also be available after the workshop for this one. And so the online workshop series is a monthly event. So please tune in for future online workshops. You can follow AG2PI on Twitter or join the AG2PI or Cyverse mailing list. So there will be more workshops on various topics in the coming year. So in this workshop, there are 3 main sections. The first one, we will be covering the basic theories behind the genome-wide association study in a very broad stroke. And the second part, we will go through the process of how to conduct GWAS experiment, what things to consider, and what are the resources to do so. And last, there's a hands-on session using maize and sorghum data as examples to demonstrate how to conduct some basic analysis with GWAS. So for this part, I've already put together some example data and scripts on this link, my GitHub repo. So yeah, I see that Ryan has put the GitHub link in the chat channel. So there are some software and R package to be installed if you want to try the script. So we'll go through that part later. And since we're talking about the chat channel, so if you have any questions during the workshop, you can type up in the chat channel. And also I'll make a stop every few slides. And if you have any questions or comments, you can also speak up. All right, without further ado, let's get started. Okay, so the topic of this workshop is genome-wide association studies. So GWAS is an observational study to dissect the complex genetic architecture of complex traits by testing the associations of variation of genetic markers with the variations in the phenotypes across an assembled population. So the concept of GWAS was invented by researchers in human disease genetics back in the mid-1990s. And the first GWAS paper was published in 2002. And since then, as you can see from the top panel here, the number of journals and the number of case studies about GWAS increased dramatically in human research. And this is also true for plants. So the first GWAS paper published in plants was back in 2005. So that's like 3 years after the first publication in human. And it was done in Arabidopsis. So the bottom panel here is an example of maize. So since 2009, you can see a trend of dramatic increase of number of publications on maize. So each kernel here is a publication of a trait in maize. So over the years, we've seen the increase of the popularity of genome-wide association studies. So the aim, or the major aim of genome-wide association study is to find the variation of the genetic markers with the variations in phenotypes. So there are four main key factors that drive the development of GWAS. So these factors are germplasm or population. So when we talk about genome-wide association study, we always talk about that in the context of a specific population, mapping population or diversity panel. And the second key factor is genetic markers. The third one is statistical model, and the fourth one is the phenotype. So this diagram here is the framework we're gonna use along our workshop today. And we'll go through each of these key factors one by one in the following presentation. But first, let's take a brief look back of how did we get there, because genome-wide association study was developed in back in the 1990s, but there are researchers of mapping genes underlying quantitative traits much earlier than this. So there are two direct precursors of genome-wide association study. So they are linkage analysis and association mapping. So these are two complementary methods. So let's talk about the linkage analysis first. So linkage analysis for quantitative trait locus mapping, or QTL mapping, is a statistical method for discovering the locations of loci underlying a trait by testing for the co-segregation of genetic markers with the traits. So this is usually done with experimental crosses. So it could be done with biparental population. For example, you can choose two parents and cross them, and you have an F1 hybrids, and you cross that F1 for one more generation, you got a F2 segregating population. Or you can self the F2 for several more generations. So in this case, you will get a reals or recombinant inbred ones. So this type of population are these type of populations are completely controlled genetic background population. So to do the genetic mapping with this kind of populations, so a lot of statistical models were developed, including single marker analysis, interval mapping, composite interval mapping, etc. So you don't need to know the details of all these models, but the point is that these statistical models directly inspire the GWAS models that we'll be talking about later. So the complementary method of linkage mapping is association mapping. So this is a statistical method identifies QTL by examining marker and trait associations that can be attributed to the strength of linkage disequilibrium, or LD, between the markers and the functional markers in the population. So instead of creating the population, the association mapping utilize natural population. So the researchers go out and collect the existing lines that has been out there for a while and take the advantage of historical recombinations in the population. And depends on the types of marker you're using. The association mapping can be categorized into candidate gene association studies and genome-wide association studies. And we'll talk about these two types of association studies later in the slides. So like I just said, the LD or linkage disequilibrium is the key for association mapping. So let's take some time to look at the LD. So the definition of LD is the non-random association between two or more loci in a population. And this is caused by a lot of genetic factors, including selection, recombination rate, mutation rate, genetic drift, etc. So an example here are illustrated with these dots. So there are 4 types of dots here: the blue ones, the black one, the white one, and the red one. So each of these are an allele. So if the population is under linkage equilibrium or under Hardy-Weinberg equilibrium, the association of these alleles are completely random. For example, the chance of the blue dots and the black dots, the blue dots and the white dots, the red dots and the black dots, and the red dots versus the white dots, the chances are equal. So they're completely random. But if the population is not under Hardy-Weinberg equilibrium, you will see that, as illustrated here, the chances of the blue allele appear with the black allele, and the chances of the red allele appear with the white alleles are much higher than other cases, like the blue allele versus the white allele. So this is the phenomenon of LD. So in general, the strength of the strength of this kind of correlation of 2 alleles, or the association of the 2 alleles, is a function of the distance between them. So the closer the 2 alleles or the genetic markers, the stronger the LD. But this is not always the case. There are exceptions. But with this phenomenon, we can use this to do genetic mapping. So for example, if there's a causal variant in the candidate gene, and we are not able to genotype it, but if there is a SNP marker that is in LD with this causal variant, and we can genotype this marker, and this marker is associated with the trait we're interested in, then by detecting this association, we can map to this genetic interval, genomic interval that contain this candidate gene. So this is how LD works in association study. So one of the example here is, so we're using this Berry number as traits. So there are 3 SNPs demonstrated here. So the causal SNP is the highlighted one. So we know that this SNP is underlying the phenotypic variation of this trait, but we're not able to genotype this one. So there are 2 markers around this causal SNP. One is in low LD with the causal marker. One is in high LD. So in high LD means that these 2 appears together in higher chances, like the blue alleles and the black allele here. So since we're not able to genotype this causal allele, if we can genotype this marker that is in LD with it, then we can test the significance of association with the trait. And if it's significant, then we're able to map the genomic region contains this causal marker. And this is how we utilize the phenomenon LD to conduct association mapping. So there are multiple statistics to characterize the LD in the population, and there's no single best statistics. I left a link with a slide deck of how those statistics are calculated. So if you're interested, so you can check those slides after this workshop. Oh, and I'll upload the slides to the GitHub after this workshop as well. So let's get back to association mapping. So in earlier slides, we mention that there's two types of association mapping. So one of them is candidate gene association mapping, and the other one is genome-wide association mapping. So candidate gene association mapping is a hypothesis-driven method. So it needs prior knowledge about the location and the biomedical or the regulatory pathway about the candidate genes. So for example, if you are studying a trait and you believe that a set of candidate genes is involved in the control of the trait, then you can just genotype markers that are around these candidate genes and do association and find out which of them is associated with the trait you're interested in. So by doing this, you will miss the unknown loci associated with the traits because you're not investigating the whole genome. So, and also this method is highly trait specific. So if you change a trait you're studying, then you need a new set of candidate genes. And but by doing this, the cost is relatively low because you only need a smaller size of population and a smaller set of the genetic markers. So in earlier days, in maize, for example, to conduct candidate gene association study, people use less than 100 lines or 100 genotypes with 100 of SNPs. So it's relatively cheap. And with the advance of next generation sequencing, we're able to genotype the SNP markers across the whole genome. So then there's this genome-wide association study. So genome-wide association study or genome-wide association mapping is a comprehensive approach to systematically search the genome for causal genetic variants. So by doing this, there's no prior information about the candidate genes are required. So all you need to do is to assemble a panel, and you genotype the individuals with genetic markers, usually SNPs, and you collect the phenotypes you're interested in, and you select the appropriate models. Then you can do the genome-wide association study to find the potential markers and candidate genes underlying these traits. But of course, this method is of higher cost because usually you need the community's efforts to assemble a panel that is much larger in the sample size. And also, you would need more markers to characterize the historical recombination events. So it's more expensive than the candidate genes association mapping. So again, I want to make the point that linkage mapping and association mapping, these two direct precursors of GWAS, they are complementary. So both of them are powerful tools in geneticist toolbox. So often that the pros of one method is the cons of the other methods. So for example, for linkage mapping, let's take a look at the cons of this method. So usually people use biparental populations, sometimes multi-parental populations. So you make the experimental crosses and you create the recombination events yourself. So first of all, that's going to take a long time if you want to self the F1 for several generations. So that's take a long time to make the population. And since you only have like 2 parents, the diversity for alleles are relatively low. And since you only self them for several generations, the combination events are not a lot, and that will give you a lower mapping resolution. But for association mapping, you don't need to make the cross yourself. You only need, so all you need to do is go out and collect the lines that are existing there. So those lines have been out there for a while. They can be land races, they can be the results of the breeding programs by the breeders. So there are a lot of historical recombination events there. So these historical recombination events can give you higher mapping resolutions. And of course, the diversity for alleles are much higher than those you created by making the crosses. But the association mapping, So it's an observational experiment. You collect those genotypes and you phenotype them. So maybe you're only interested in 1 or 2 traits with this population, but there's a substantial amount of variation in many other phenotypes as well. And you need a larger population size for association mapping to detect QTL. And of course, you need a genome-wide marker of higher density, and more validations are needed for the candidate genes. But for linkage mapping, you only need a relatively small population size. Usually for biparental population, let's take recombinant inbred lines, for example. There's usually the scale of 100 lines you need to do the QTL mapping. And you don't need that many SNP markers or other type of genetic markers because there's not that many recombination events that you need to capture. And once you locate the genomic interval in linkage mapping, you can go through the process called fine mapping to further pinpoint the real candidate gene underlying these traits. So these are the pros and cons of the 2 direct precursors of genome-wide association study, linkage mapping and association mapping. Yeah, so this is a brief history of genome-wide genetic mapping. So are there any questions so far? So you can speak up or type in the can we perform GWAS with traits with no heritability? You can, but probably you won't find any meaningful hits.
I have a question, sorry.
Yeah, please go ahead.
So since we are trying to find the genomic regions that cause or that are associated with a specific trait, how important is the relatedness of the population? If maybe, I mean, I'm talking about a breeding population that is not necessarily in diversity panel. It's just like a breeding population that are highly related. So how important is it to consider to do GWAS?
Yeah, that's a great question. And we will be covering this in more detail in later slides. The short answer is it's really important. The choice of the population, whether it's a diversity panel or it's a result of the breeding program, that's one of the deciding factors of the successful of your GWAS experiments. We'll cover that in detail in later slides, but thank you, that's a great question. Sorry, any other question or comments? If not, we're gonna go on with the second section of this workshop. So sorry, it's still the first section of the workshop. So we've been talking about linkage mapping and association mapping. So those are the direct precursors of genome-wide association study. So now let's focus on genome-wide association study itself, and let's see how this method evolved over time. So when we talk about genome-wide association study, let me remind you again, we're gonna use this framework. Consists of 4 key factors: germplasm, variation genotypes or genetic markers, variation phenotype, and also the association models. So let's start with the model. So the most naive model for genome-wide association study is essentially t-test. So let's take a look at the example here. So say we have a panel of individuals and we genotype them with SNP markers across all these locations. And the phenotype we're interested in is plant height. And there's variation of plant height among all these individuals. So the most simple way to test if a marker is associated with this trait is to conduct t-test. So let's take a look at this marker. For example, there are 2 alleles, A and G. So we want to compare the individual carrying the A allele versus the G allele. We want to compare the mean of the plant height of these two groups. And if the t-test, if the result is significant, then we can say that, okay, this marker here is associated with plant height. And we can go through this process for all the markers, all the positions with genotypes. So there are two major problems with this naive model. The first one is the false positives accumulates across a large number of markers. So if we look at a single locus here, so let's say we set the significant level equals to 0.05. So 0.05 is the chance that we found false positive here. And that is not very high for a single locus. But if we have a million markers or tens of thousands of markers, and each of them are assumed to be independent, the number of false positives could accumulate, and that could end up with a lot of false positives. So that's one thing. And the other thing is, we're talking about genome-wide association study within the context of a germplasm or a population. So there's a genetic background of the specific population you're using. So that relates to the question just asked a few slides ago. So this naive model of t-test, it does not address the relatedness among the individuals in this panel. So when we talk about the relatedness among the individuals, usually we're talking about two things, and we'll go through them in the next couple of slides. So first thing about the relatedness among the individuals is called population structure. So let's take a look at this example here. So this Morris et al. 2013 study, they collected over 900 lines of sorghum so that's a worldwide collection. And you can see that there are a lot of subpopulations here. There are sorghums from, say, West Africa, East Africa, India, China, etc. So the allele frequencies between these subpopulations could be systematically different, and this can lead to false positives in association study. Let's go through a hypothetical examples of why this could cause false positives. So with this example, we have two subpopulations. One subpopulation are the sorghums from West Africa, and the other subpopulation is the sorghum from East Africa. So let's say, hypothetically, the genotypes from West Africa are taller than those from East Africa. And the other trait is the resistance to a disease. So the genotypes from West Africa are more susceptible to this type of disease, while the genotypes from East Africa are more resistant to this type of disease. And this is caused probably by the geographical and climate distance of these lines, or the familial relationship, etc. And there are two SNPs here. One SNP called the SNP1 is underlying the trait plant height. So there are two alleles for the SNP A and G. So the West Africa lines are enriched with the A allele, and the East Africa subpopulations is enriched with G allele. And if you conduct t-test, you will find that this SNP is significantly associated with plant height. And we know that it's underlying this trait. And there's another SNP called the SNP2. So this SNP is underlying the trait of disease resistant. So the C allele is more enriched in the West Africa lines, and the G allele is more enriched in the East Africa subpopulation. And now, if you're only studying the plant height, you want to conduct GWAS for the trait plant height using this population containing these two subpopulation. You would go through all the markers and see if the marker is associated with plant height, this trait. You will find both of the SNPs are associated with plant height. But in reality, only SNP1 is underlying these traits. SNP2 is actually underlying the disease resistance. And this is how population structure could lead to false positives or type 1 error. The other layer of relatedness among the individuals is called kinship or unequal familial relationship. So this could be caused by recent co-ancestry among the individuals usually exist in a panel. So an example here would be, for example, this green box is a population we collect for genome-wide association study. And maybe in this population, there are some offsprings of some certain combinations of parents in this population. So if this exists, then some of the alleles, like the combinations of alleles, would be fixed in this population. So in this example, the small a and capital B are always together. So this could cause false positives, just like the example we just used for population structure. So because since these two will always be together. So to control for this, so people usually use kinship matrix in the model, which will be covered in later slides. So these are the two major relatedness among the individuals. Do you have any questions so far? Or comments about kinship versus the population structure? Because these two are really important for genome-wide association study. And the control for these two factors was a major advance in the model in genome-wide association study. And we will go through how to control these two types of relatedness in the later sections. Okay, let's move on. So after people realized that there's this multiple levels of relatedness among the individuals, this unified mixed model framework for association study was developed. So in this model, you can fit the SNPs, the population structure, the kinship into a mixed model. You can fit the population structure as fixed effect and kinship as a random effect. So by fitting these factors in the mixed model framework, you can control for the relatedness that could lead to false positives in the model. So this mixed linear model framework was published in 2006. Okay, we'll talk about questions later. So from the simulation study here, you can see that so the upper panel of the figure is showing that the different types of models. So the Q plus K means the mixed linear model that control both population structure and kinship. And there's also the Q model, the model only control for population structure, and the K model, the model only control for kinship. And there's a simple model, which is basically t-test. So the y-axis here is the cumulative p. So this is basically a scatter plot of expected p-value versus observed p-value. So in a perfect case, if there's no false positives, it should be a diagonal line. So you can see that for all the traits here, the flowering time, year height, and year diameter in maize. So these are 3 traits with high population structure, moderate population structure, and low population structure. You can see that the Q plus K model control the false positives the best compared to other types of models. And the lower panel of this figure, the y-axis here is the adjusted average power. So this is to measure the ability of the models to detect true positives. So the higher the curve, the higher the power to detect the true positives. And again, across these 3 types of traits with various levels of population structure, you can see that the Q plus K models always perform better than the other types of models. So this simulation study demonstrates that by including population structure and kinship in the mixed model framework, we can increase the ability of finding true positives while reduce the chances to find false positives in genome-wide association study. And this is how population structure are addressed in genome-wide association study. Okay, let's take a look at the chat. Does kinship relate to, say, phylogenetic of genes, or it's mostly a function of SNPs? I would say both. So you can calculate the kinship based on a subset of SNPs, and also you can calculate them with a genome-wide set of SNPs. So usually people in earlier days, when there's not that many SNPs are readily available, they use a subset of SNPs. But later, especially when these Q and K models were brought up, people use a genome-wide set of markers to calculate for the kinship. But in later development of the models, so in order to increase the speed, because the calculation of the kinship are the pairwise correlation of each SNPs. So if there's a large set of SNPs, that would take a lot of time to do so. And then the researchers started to use the subset of the SNPs to overcome this problem. So yeah, the answer is you can do this with both. What is the difference between random effect and a fixed effect? Okay, so this is a hard question to answer because the mixed model is a whole new topic. So I don't think we have time to cover that. But in general, the fixed effect would be something you realize that has a effect on your experiment, and the random effects are the ones you're not sure if they're affecting your experiments. I'll include a link of tutorials about how to decide fixed effect and the random effects later into this slide, so you can refer to. Is a psi random effect or fixed effect? It's usually a fixed effect. All right, so when we include PCs as fixed covariates, also GRN can cause the double counting because yes, that's true. So, well, in general, with more factors you include as covariates, the higher the chance that you overcorrect the model, which could lead to false negatives. And we will see that in an example in later slides. So since the invention of this mixed linear model framework in GWAS, there has been a lot of improvement based on this framework. So in general, you don't need to know the details of all the models, but in general, people try to increase the computational efficiencies by creating computational shortcuts, and also try to increase the statistical powers for the mixed linear model framework. So in our hands-on tutorial session, we will be conducting genome-wide association study using this Q plus K model, or the mixed linear model, and also the one shown here with the highest computational speed and also the statistical power called farm CPU. And other than this mixed linear model framework, there are some other alternative frameworks like the Bayesian methods, like the Bayesian R, blob, the base A, base B, the base C, etc. So again, you don't need to know the details of these models. You just need to know that other than the mixed linear model, there are some other framework exist. All right, we have been talking about the germplasm, the variation of genotypes and the models. Now let's talk about the variation in phenotype. So so when we think about phenotype, usually we think about the morphological traits like the plant height, like the weight of an animal, like the flowering time, those kind of things. But actually, there are many layers of phenotype. So the definition of phenotype is much more than that. So there's also biomedical traits like the metabolome. There's also molecular traits like the transcriptome and proteome. So for example, if we use the gene expression profile of an individual as an example here, then we will be ended up doing something called EQTL analysis. So some of you are probably have heard about that. So the point of this slide is to demonstrate that we can expand the list of phenotypes by investigating multiple layers of the genotype we're observing. So a quick summary for the 4 key factors that drives the development of GWAS. So when we talk about germplasm, we started with biparental population and multiparental population with linkage mapping. And later there's association study, candidate gene association study, and the genome-wide association study. Usually we use a diversity panel. And also there's for the variation in genotype. So earlier days, people use like RFLP markers, microsatellites, SSR, and recently the SNP markers. And for the models, the naive model is essentially t-test. And then there's general linear model, mixed linear model, and the improvements based on the mixed linear model framework. And also there's some alternative framework for the association model. And for phenotype, there's agronomical traits or morphological traits. And there's also the biomedical traits and traits at other levels we should consider. Okay, so this is a summary so far. So this is the summary of our first section. And in these slides, we'll talk about some challenges about GWAS. Are there any questions so far? All right, so if not, let's move on with some challenges of genome-wide association study. So for genome-wide association study, so one of the challenges, how do we deal with the loci with low minor allele frequency? So the low minor allele frequency are usually, so they usually have a smaller p-value in association study and the higher chances of being false positives. But these rare alleles could be causal, but they're of lower confidence. So, so this is one of the major issue of GWAS, whether we should consider this these markers, or we should exclude them, or filter them out. And the second challenge of genome-wide association study is how should we establish an appropriate significant threshold for the model? So, so there are 3 major types of threshold. One is bound-friendly correction, which is overly stringent, and the other one is false discovery rate. So this is less stringent, but it's also under the assumption that all the markers are independent, which is often not the case in genome-wide association study because the phenomenon of LD. And last, you can do permutation study, permutation test to establish the threshold, like the popular methods in QTL mapping. But this is very computational intensive. And we will go through bound-friendly correction and false discovery rate in later slides. And the other major problem is called synthetic association or ghost association. I'm not sure if anyone of you have heard of this. So it means that the causal genes or the causal SNPs underlying the traits are sometimes located away from the GWAS peak you found. So this is an simplified example from the sorghum shattering 1 gene. So the causal polymorphism are this presence and variance variation, and also this mutation affect the splicing in the gene. But these two cannot be genotyped. And this SNP near this gene can be genotyped. And by performing a association, this SNP is actually detected as the SNP that passed the threshold we set up. But the real causal SNPs are away from this peak. So this is the phenomenon of synthetic association. So the take-home message here is that when you find a GWAS peak, it's not guaranteed to be the causal SNPs. So when conduct genome-wide association study, you should keep that in mind.
I have a question.
Yeah, please go ahead.
Yeah, the question on the minor allele frequencies. I know it's still controversial, but so what is the standard? Are we doing where the minor allele frequency is less than 0.05 or 0.01? What is the general literature and standard in the industry?
Yeah. Yeah, that's a good question. So the short answer is that there's no gold standard for that. So the convention in earlier days was to filter out the markers with minor allele frequencies less than 0.05. And it also depends on the size of your population, because 0.05, so if you have a panel with over 1,000 genotypes, that would be a lot of individuals. If you have a panel of smaller set of inbred lines or genotypes, say 100, then that would be only a few individuals. And so I'll show an example in later slides to show that actually these rare alleles, the markers with smaller minor allele frequency, usually less than 0.05, they usually have larger effects and can be causal genetic variants underlying a lot of traits. So currently, for genome-wide association study, so I've heard about this opinion from some experts saying that they would recommend you to keep those markers with low minor allele frequencies. And you can see if they appear in the final association. So if the p-value are really small, then you can check if it's caused by the small minor allele to check the actual biological meaning of that marker. So right now, people tend to keep those markers with low minor allele frequencies compared to the earlier days where people just filter them out from the start of the analysis. Any other questions about this? Okay, so that was about the challenges of genome-wide association study. Now we'll spend a few slides on the future of GWAS. So far we've been talking about the variation in genotype. So we've been talking about that on a DNA level. So that is the polymorphism of the DNA, like the SNPs. But actually, there's much more layers of that than genome, right? So there's this epigenome, there's transcriptome, there's proteome, metabolome. So each of these could serve as markers to associate with the variation in a phenotype. So for example, right now, there are a lot of studies using a method called transcriptome-wise association study, or GWAS. So instead of using the polymorphism of SNPs, the polymorphism of DNA, like SNPs, to associate with the phenotypic variants, this method used the gene expression level as explanatory variables to associate with the variation of phenotype. So each dot on this Manhattan plot here is not a SNP like GWAS. It's actually gene because it's the expression level of these genes that were used for the association study. The same concept can be applied to all these levels, like proteome and metabolome. So there are more and more this type of association study that is being developed right now. And the other thing is, so far we've been talking about calling the SNPs or other types of markers using a single reference genome. But with the advanced in the sequencing technology, so there's more and more pan-genome for some species right now. So for example, this paper published, I think probably last month. So they sequenced, they assembled this 33 rice genomes and created a pan-genome for rice. So with the pan-genome, you can use markers such as structure variance and also variation, and also the SNPs that you won't find with a single reference genome. And this type of pan-genome have been done in soybean as well, and also for maize. Like I know that one publication is on the way for maize, the NAM parents, the nested association mapping parents. So they sequenced about 25 maize genome lines and created a pan-genome for maize. And by using pan-genome as reference genome, we are able to detect more genetic variants to associate with the variation in phenotype. And the last thing I want to talk about is high throughput phenotyping, or phenomics. So we talk about the expansion of the list for phenotypes. So with the new phenotyping technology, like using UAVs or drones or all types of cameras or these new sensors, we will be able to further expand the list for the phenotypes. So instead of just measuring the trait at a single time point, we can phenotype the same traits through the developmental stages. So that would give us some insights into the dynamics of the traits. So this is also one of the future directions for genome-wide association studies. All right, so that concludes our first section of this presentation. So any questions so far? You can. How low is low minor allele frequency? Like I just said, there's no gold standard for that. So usually people use 0.05 as the definition of rare alleles, but that also depends on the size of your population. For a significant SNP, how will we expect this minor allele frequency? I'm not sure I understand this question, because when you use the SNP as the input, you include those in the association model. You already know the minor allele frequency for that SNP. I mean, if you can clarify your question, then we can probably talk about that later. All right, let's move on to the next section. So let's walk through the process of how to conduct genome-wide association study. So from how to select for population, how to consider the genetic marker, how to address the population structure, how to deal with the phenotypes. So let's walk through the whole process. So the first thing to consider is the species and the germplasm. So when you choose a population for your GWAS experiment, you need to carefully consider all the genetic aspects and the resources available for this panel. Like how was the panel assembled and why was it assembled? Like how diverse is the genetic, so what's the genetic diversity of the panel, and what's the genome-wide LD look like, and how's the population structure, and are there a lot of genetic marker available for the panel, and do you want to use the whole panel or just a subset of that, and how many marker are you going to use? So one example I want to show here is also using maize as an example. So the above panel here is a panel called Wisconsin panel. It has 501 genotypes here, and it has several subpopulations, including stiff stalks, non-stiff stalks, popcorn, sweet corn. And the bottom panel here has 575 genotypes. So it's even larger than the Wisconsin panel. But all of these are collections of popcorn. So if you want to study popcorn-related traits, then probably you should go for the second one. But if you're not studying for that, you probably would want to go with the upper one here. So that is one thing to consider when you choose the panel you work with. And the other thing you need to consider are like the size of the panel. So for example, there's this Ames panel here for maize. So it is a worldwide collection of more than 3,000 inbred lines of maize. And most of them, 2,800 of them, have been genotyped with GWAS. And the LD decay is pretty fast for this panel. So it reaches 0.01 at about like 1 kb. And some of the traits have been phenotyped, like the kernel color, the flowering time, the plant height. So when you consider about your own experiment, will you have the resource and the time to grow all these 3,000 lines and phenotype them? Because this is a huge panel, and it has this crazy population structure here. Or you can start to work with a smaller panel, but also very diverse. So this is an example of MAZE2. It's called MAZE282 association panel. So this collection of lines are biased towards the temperate lines, but also it's a collection through the world. And this panel has been used for numerous mapping studies. So on this one website, Panzea, it has 57 traits for multiple environments. So this is a very valuable resources if you want to start your own experiment. And this panel has been genotyped comprehensively. So all of the lines in this panel have been genotyped with whole genome sequencing. So that has more than 83 million variants. And there's also RNA-seq data from 7 tissues for this panel. So unless you want to generate the markers yourself, or you want to do like RNA-seq or other types of sequencing yourself, you would probably want to start with this MAZE282 association panel compared to other panels with less marker or other types of data available. So this is something to consider when you choose your panel for the species you're working with. The second thing is the marker. And again, I would recommend you to check the videos from previous workshop by Dr. Landis about the SNP data analysis. So there are all types of technology that can generate SNPs you can use as markers. There are SNP arrays, there are GWAS, there are RNA-seq, whole genome sequencing, and each method you choose will have a corresponding bioinformatics software or pipelines for the processing of the SNPs. And also the cost of these technologies varies, and also the coverage of them are not all the same. So this is something to consider. So once you have a population and also the types of markers, the next thing to consider is the field design and the phenotyping. So the accurate phenotyping is the key of GWAS, because if you're not assemble your own population and genotyping them, then the only thing you can consider is the accuracy in the phenotyping. So when you design for your experiment, you would want to use techniques such as the block design and randomization, and also you want to use biological and technical reps to increase the accuracy of the trait phenotyping, and it would want to include checks across the field to provide information for later spatial corrections. Because as the figures show here, the spatial correction could improve your trait, the phenotyping results dramatically. So this is something to consider when you design your field experiment for your genome-wide association study. And the last thing I want to emphasize about phenotyping is to document well about your phenotype. Like what's the definition of your phenotype? What exactly did you measure? So you can take photos if needed, or videos if needed. This is useful for your downstream analysis, for your modeling process. And also it's very useful for other researchers to reuse your data. So once you get once you finish the phenotyping, so the next thing is to get the phenotype right. So first you want to conduct some exploratory analysis by plot your phenotype and check the distribution of the traits. If it's normally distributed, or it's left skewed, right skewed, etc. And if it's skewed, then you might want to consider some transformation techniques, like take log transformation or Box-Cox transformation. And if there are any outliers, you want to check what's happening there. And as we just talked about, the factors in the experimental design, like the reps, the treatments, the years, the environments, you want to take these into the consideration too. So one technique of doing this is called best linear unbiased prediction. So I left a link of tutorial of how to do this, so you can check it if you're interested. And with this method, you can also estimate the how heritable is your traits. So I got a question earlier about if we can do GWAS for traits with zero heritability. So the answer is you can try, but it is very unlikely you will find meaningful association signals, because there's no genetic components in the trait. Can you please send a link in the chat? I'll upload this slides to the GitHub after the workshop. The next thing is to get the marker data right. So unfortunately, there are many formats of SNPs. So there are HapMap formats, there are VCF, variant call format, there are PED and BED, and there's other. So you would want to convert the format of the SNPs to the one that is required by the software you're using. And there are softwares to do this, like Tassel, like VCF tools, G tools, etc. Yeah, this is also something you want to keep in mind. So let's talk about the filtering of the markers. So we were talking about minor allele frequencies. So this figure shows that in many traits, so this is from a human disease example. So the rare alleles, so the markers with very low minor allele frequencies, they usually have higher effect size. So this could be false positives, and this could also be some underlying biological process there. So so you don't want to exclude them at first. So so if those were detected in your association study, you can look into it and see if it's really try to explore if it's really causing this this variation in your phenotype. So like I just said, there's no gold standard for filtering out the low minor allele, the alleles with low MAF. So usually people, sometimes they use 0.05, but this is actually kind of arbitrary because it really depends on the population size you have. And also you would like to filter for the missing rate and the heterozygosity rate, because even if you're using inbred lines, there's still a phenomenon called the heterozygosity residual. So that means some part of the genome are still heterozygous. So those can be removed or can be dealt with in other ways. But that's something to consider for your marker data. Okay. So the next thing is to address for the relatedness among the individuals.
I have a question, sorry.
Yeah, please go ahead.
Can heterozygous markers be also associated with some genetic phenotypic traits or not? Like,
yes, they can.
They can be.
Yes.
So, what would be the step to recognize them if you are filtered them out?
So usually in the GWAS model, so the model won't care if it's heterozygous or homozygous, like if it's AA or AG or GG. So in the model, usually it's numeralized. I would say that if you're using a panel of inbred lines and there's unusual high percentage of heterozygous sites, you will need to start to pay attention to if there's any, some errors in the genotyping process. If it's of a very low percentage, then you can just keep them.
Thank you.
Yeah. All right, let's move on with the address the relatedness among the individuals. So we were talking about population structure and kinship, and how the mixed linear model framework deal with these two types of relatedness among the individuals. So in the model, so people usually use two ways to control for this. One is to use the output of the structure software. So it can be applied to multiple types of markers like microsatellites, alpha-LP, and SNPs. And also there's this new version of the software called Fast Structure that can deal with a large SNP sets. So it's pretty fast. But the more common way to control for the population structure is using principal component analysis, or PCA. So usually we include a few first few principal components that can explain a large percentage of the variation in this population. So one of the way to decide how many PCs to include is to use this scree plot for PC. So the y-axis is the eigenvalues, and the axis is the number of PCs. So what people usually do is to find these turning points of the eigenvalues, or the elbow of this. So you can see that from this example. So after the first 3 PCs, the eigenvalue kind of plateaued. So in this case, we can only include the first 3 PCs into the model to control for the population structure, because the marginal effects of including more PC would be marginal. The marginal effect of including more PCs would be not very helpful in this case. And to address the kinship, usually people use kinship matrix. So kinship matrix can be calculated with identity by descent, or IBD kinship. So this is usually you have a clear pedigree of your population. So probably in animals, this is easier than in plants, because in plants we don't usually have a clear pedigree information for the panel we're working with. So instead of using the pedigree information, we use something called identity by state. So the way to do this is to compare 2 or more individuals with all these loci has been genotyped. So if they share more alleles in each locus, then we can estimate that the more allele they share across all these loci, the closer the relationship of these individuals. And there are multiple algorithms for estimating kinships. And I also include a link for the way to calculate the IBD and IBS kinship matrix here. And you can check this after the workshop if you're interested in this. And then is the choice of GWAS model and software. So there's really over 100 free softwares and tools to conduct GWAS with. And there's also multiple choices of models as well. So, so there are several factors you should consider when you choose this. Are you familiar with command line, or you're more familiar with the graphic user interface? So there are softwares that can do both, like Tassel, and there's also some online portals for you to conduct genome-wide association study by dragging and clicking. So like the Cyverse discovery environments, like this Easy GWAS portal and GWAS-PP. So these two were designed for Arabidopsis. And for command line tools, you would have more choices. So there are software written in C++, Java, which are pretty fast. And also there are the ones written in R. So that's more intuitive, but slower. So in the later hands-on section, we will go through an example using the Cyverse discovery environment. And also in the GitHub repo, I uploaded. So there are scripts for using Tassel and Gapit to conduct genome-wide association study with command line. So you're also welcome to try that later hands-on section. So the next thing is the choice of GWAS models. So we've been talking about this mixed linear model framework and the improvements based on this framework. And there are also alternative frameworks like the Bayesian method. So when you consider which method to use, you should consider the species you are working with and the population you have, and like the ideal statistical power and what kind of computational resources you have. And if you have any prior knowledge to the traits, like if this trait is of higher heritability or it's of lower heritability. So as shown in this simulation study across multiple plant species, crop species, so with simpler genetic architecture, like they simulated 16 QTN underlying the traits to 256. So you can see that usually the Bayesian method perform better than the other ones. And then there's this farm CPU we'll try in our hands-on session. And the baseline they used here is a mixed linear model. But since you're on these steps already, so you've already gone through choosing a population, get the genotype data, phenotyping, and all the modeling to correct for phenotype and filter for the genotype. You can try multiple models and see which one give you more reasonable results. Because compared to previous steps, trying all kinds of models are actually cheaper than the previous steps. So the typical outputs of GWAS are tables like this. So you would have the ID of your SNP markers, the genomic position of them, the p-value, the minor allele frequency, and some other model parameters that outputs from the model. And also some software will calculate the allele effects, like how large is the effect on the traits you are studying. So in the hands-on section, I included a script that allows you to take the output and visualize them, which is something we'll talk about right now. So the most iconic plots for the results of GWAS is this Manhattan plot. So it is essentially a scatter plot used to show a large number of SNPs and their significance. So the x-axis of Manhattan plot are the genomic positions of these SNPs, and the y-axis is the negative logarithm of the association p-value. So depends on which model you're using. The Manhattan plot can look a little bit different, but the information they're showing are essentially the same. So I know you're probably more familiar with this kind of Manhattan plot with its peaks or towers, but that's not always the case. So it really depends on the method you're using. Let's take a look at the question now. Which is the better platform to use, command line or graphic interface if you're using millions of SNPs? The command line. It will offer you more freedom of operating those high dimensional data, and it's much faster than the the graphic interface. Often the larger the data set, the more likely it is to command. Yeah, that's true. And the other we have seen is called QQ plot or quantile plot. So this is also a scatter plot. So the way it is constructed is you calculate the p-value from the GWAS model and you rank them from the largest to smallest, and you check the distribution of the observed p-value versus the expected ones. Because the expected ones should follow a uniform distribution, because the null hypothesis is marker is not associated with the traits. So if you see something like the blue dots, that's probably a good sign, because there are some SNPs with p-values that higher than expected. So this could be, especially the spread out, could be a significant hit. And if you see like situations like the yellow dots here, it's aligning with this expected lines, then probably it means that there's no significant hits at all. And if you see the green dots below the red line here, it probably means that it's overcorrected, like you included too many covariates, or you don't have enough SNPs to cover the genome. It could be a lot of reasons. So QQ-plot is an essential tool for detecting problems in a GWAS. And then the thing I want to talk about is how do we decide a reasonable cutoff for the significance of p-value. So like we mentioned in earlier slides, so this multiple testing problem could be a huge thing. So let's assume that we have n number of SNPs. So so for each SNPs, we would set a significant level for type 1 error, for example, 0.05, and assume that all of them are independent. So that is fine for one locus, because there's only 5% of the 5% of the chance that the association you found are false positive. But if you have 1 million markers, and these are assumed to be independent in the model, then you could end up with more than 50,000 false positives, and that's just not acceptable. So to address this multiple testing problem, people usually use three methods: the Bonferroni correction and the false discovery rate, and also the permutation test. So let's take a look at the first two. So for the Bonferroni correction, so we mentioned in earlier slides that it's overly stringent. So let's take a look at why it's overly stringent. So so for Bonferroni correction, it sets a desired type 1 error level alpha, and the corrected level would be alpha divided by the number of tests you perform. So if you set this alpha equals to 0.05 and you have 100,000 SNPs, then the p-value here is really, really small. So only the SNPs with p-value smaller than this could pass the genome-wide threshold. And the other, so the alternative way to do this is false discovery rate. There are multiple ways to do this. So there's this BH procedure and BY procedure, and a lot of variants. So this is so the common cutoffs for FDR are 0.05 and 0.1. So we're not going to go into the details of how FDR is calculated, because it's a little bit less intuitive than the Bonferroni correction. But these are the ways to address multiple testing problems. Let's see. What if you find a lot curve lines like this? Okay, we'll talk about that later. Okay, so the last thing I want to talk about is, so now you have gone through like all the steps, and you found the significant SNPs that pass the threshold you set. The next thing is to find the candidate genes. So you can search for candidate genes using local LD versus genome-wide LD. So for local LD, an example is that you have a blow up of the region, and you calculate the pairwise LD in this region, and you can look for candidate genes in this. But if you don't have a high density set of SNPs, the local LD could be really noisy. So an alternative for this is to use all the markers you have across the genome to calculate a genome-wide average LD. So this would be less noisy. The way to do this is to, when you got the genome-wide average LD, like this example for Ames panel, if it's 1 KB, then you can set up a window centered on the SNPs and look for the candidate genes in this region. And once you're in this step, there are multiple ways to go. So you can do fine mapping with some of the parents to make experimental crosses, but that's kind of time consuming. And also you can look for evidence from other layers. Like you can check the expression patterns of the candidate genes in the tissue that is related to the phenotype you're interested in. So for example, if you're studying leaf-related traits in plants, then you can probably check the candidate genes in that region if some of them are highly expressed in leaf tissue. And that could be some evidence for you to follow up. And also, if you have RNA-seq data for this panel of lines, then you can construct a co-expression network. If some of the candidate genes are co-expressed with a known gene that affect these traits, then also that's higher chances that this gene is also underlying the trait of interest. Yeah, I think I'm gonna stop here. So in this workshop, we've briefly gone through the history and the evolution of GWAS. So we started with linkage mapping and association studies, and we went through the 4 key factors in GWAS: the germplasm or population, the genetic markers, the statistical models, and phenotype. And we spent a lot of time on why and how to address for the relatedness among the individuals in the panel with this mixed linear model framework. And we walked through the steps to initiate a GWAS experiment, like the choice of the panel, how to deal with markers, something to pay attention about field experiment and phenotyping, and the choice of GWAS models, and how to interpret the GWAS results. And this concludes my presentation for today. I would like to thank Dr. Pat Schnabel, my PhD advisor, and also the PI for the AG2PI initiative project, and Dr. Eric Lyons, Dr. Ryan Bartam. Ryan, how to pronounce your last name? I'm sorry. And for Dr. Cole Scott to set the workshop up. And also I'd like to thank Dr. Tintin Guo, who is a postdoc at Iowa State, for her feedbacks on the workshop. And also thank AG2PI and Cybers for the support, and all my lab members here at Iowa State and also in China Agricultural University.